Okay, we're going to go ahead and get started now. I even brought my backup brain with me, you can see. So, we're going to continue our discussion on chapter 11, finish chapter 11, and go into, actually go into the brain and spinal cord. We were talking about neurotransmitters. That's where we'll pick up right about acetylcholine. Anybody have any questions on anything before we start? Yes.
When is the lecture test?
When we finish chapter 12.
Like this coming lecture test.
The test for unit 3 or unit 4. Unit 3 is already out there.
Okay, when is it due?
Wednesday. That was not the answer you probably wanted to hear.
No,
So
it's really not.
I can probably push it back a little bit. So
How about Saturday?
just remember that the further back I push these, the closer you're going to be running into other exams.
That's true.
So just keep that in mind. Keep that in mind. So, and that's okay. Yeah, I can give you a little more time to get ready, but just remember that you're going to be, we've got 2 more exams to get done after this. So now lecture test 4 will open up as soon as I finish up on the brain. Now I expect to finish the brain by Thursday lecture. That's what I'm hoping. If not, it'll be next Tuesday. Now, the good news is the last 2 chapters are very short. So chapters 13 and 14, my goal is to finish those by Tuesday, the last Tuesday in the week, and then have a review session for the final on Thursday. So just to let you know, that's my plan. So, we'll see how close we get to it. So. But yes, I can push the test back a little further for you. Just remember that it's going to be cutting into your last 2 weeks of material, so of timing. So, okay. Any other questions? Did you have a question? Or did you? Well, don't look to like don't give me the deer in the headlight look. Because I thought you raised your hand. So.
I did, but she asked it.
Oh, okay. So, okay.
Yeah.
Anybody else have a question on anything? Okay, then I'm going to go into this and open up where we were. And because the last thing we looked at, according to the YouTube video, the last thing, that's how I find out where we stopped. I go back and check the video now. So I can't depend on my memory anymore. Acetylcholine is the most well-known of the neurotransmitters. There's between 50 and 60 neurotransmitters. Acetylcholine is the one we know the best. It's a small molecule, so it falls into the category of small molecule neurotransmitters. It's used in a variety of places. We use it at the neuromuscular junction. We just talked all about that. In muscles, we release it from the axon terminals. It drifts across the synaptic cleft, opens up the motor end plate, sodium gates. We depolarize there, and if we hit threshold, we're off and running. It's an excitatory neurotransmitter there. In the heart, it's an inhibitory neurotransmitter. When released into the cardiac muscle, acetylcholine slows the heart rate down. In fact, that's what it's supposed to do, is to slow our heart rate down. Acetylcholine is keeping our heart rate at 75 beats a minute in AP2. We'll talk a lot about the heart, but you'll see that the heart, if it didn't have acetylcholine, would be running at about 100 beats a minute. You see it here and there for right now. So that we see acetylcholine as excitatory and inhibitory. It depends on its target. The parasympathetic nervous system uses acetylcholine exclusively as its neurotransmitter. The sympathetic nervous system uses acetylcholine for its first stage neurotransmitter to release its norepinephrine and epinephrine in its second stage. We'll get into that when we get into chapter 13 and 14. So acetylcholine, we know a lot about it. We know that it has to be taken out by acetylcholinesterase or just plain old cholinesterase. It destroys the enzyme. I mean, it's the enzyme that destroys the neurotransmitter. Why do we have to get rid of it? Because we can't generate a new action potential if acetylcholine's already on a receptor site for it. If it's already opened up a sodium channel, it can't do it again until we get it out of the way and let new acetylcholine come in. So anyway, that's acetylcholine. Now there's a variety of other neurotransmitters. We call them the biogenic amines. I real honestly don't know what biogenic means, but it doesn't matter. Catecholamines. You've probably been exposed to catecholamines if you've ever smelt a raw onion. The chemical agent in the onion that makes your eyes water and tear up is a catecholamine. Now, most of the catecholamines in the nervous system involve either dopamine, which is a pleasure neurotransmitter. When we release dopamine, we feel good. Or epinephrine and norepinephrine. Epinephrine and norepinephrine are released from the sympathetic nervous system. We use epinephrine when we go into a fight or flight response. We use norepinephrine to open or close our blood vessels. The indolamines are another type of biogenic amine. Serotonin. You may have heard of serotonin. It's a mood stabilizer. Many antidepressants are built around a chemical that is called an SSRI, a selective serotonin reuptake inhibitor. In other words, a person is suffering from depression. Depression is not a character flaw. Depression is a chemical imbalance in the brain. It has nothing to do with character or personality, but it has everything to do with the amount of serotonin they're releasing into their synaptic cleft. And these SSRIs inhibit the enzymes that break down serotonin. The idea being if we can keep serotonin in the synaptic cleft longer, even if we're not releasing enough of it, it'll do its job. So a lot of your antidepressants are built around chemicals that will allow serotonin to stay in the synaptic cleft longer. So, but it is a mood stabilizer. Histamine. Who hasn't heard of histamine? Or have you ever heard of antihistamines? Okay, this is what antihistamines block the action of histamine. Histamine triggers are what we call the inflammatory response. We are exposed to an allergen like pollen and our noses run. We've all experienced that at some point, or at least we sneeze. You know, I know everybody sneezed at some point. Histamine causes our blood vessels to enlarge. It causes them to become leakier. So when we have an irritant, like in our nasal cavity or around our eyes, we release histamine from the white blood cells in that area. That causes the blood vessels to dilate. That means they enlarge. That means, and they become leaky. So what leaks out? Plasma. And as it leaks out, it stops being plasma and becomes interstitial fluid. And if it leaks out of to the outside of the body, it's now mucus or snot, you know, or watery eyes. So what do we do to stop that? We take antihistamines to block that. Now that's a localized response. We also see histamine being released on a systemic level if you're allergic to bee stings and you swell up. You're getting, it's still what we call the inflammatory response. A person who's allergic to bee stings will have a body-wide release of histamine, causing all their blood vessels to dilate and become leaky, and plasma leaks out into the spaces into the interstitial areas around the vessels, into the tissues, and we swell. We have a body-wide swelling. Of course, you're running into dying because your blood volume has dropped precipitously. Yes.
What makes a person get hives?
Again, that's another type of allergic reaction. It hives and itching and things like that all involve the release of histamine. Is it going to cause inflammation? Inflammation is increased blood flow to a region with swelling, and it's very easy to transition into hives at that point. So it doesn't always cause a tremendous amount of swelling, but it will always involve increased blood flow to a region. Even if you have a cut or a scratch, like right there, where it's red around the scratch, that's inflammation. That's a good thing. It means that we have, part of the healing process. You're sending more blood there. You're sending more oxygen there. Sending more white cells there to heal up, and that's why it's red. Red from it just because of increased blood flow. So, but yeah, it's all driven by histamine. We use all of these biogenic amines to deal with our biological clock, our emotional behaviors, fight or flight. Do we feel good? Dopamine gets us into the area of dealing with addiction with, for example, cigarettes. Nicotine released from tobacco stimulates the dopamine receptors in the brain to release lots of dopamine on that very first exposure and that very first cigarette of the day. You get this surge of dopamine and it feels good. The next puff also gives you a surge of dopamine, but it's not quite the same amount as you got a second before. And the third puff is a little bit less. And so all day long, your smoker is taking in more and more nicotine, trying to get back to that original surge of the day. And that's what creates the addictive behavior here. Okay, now, amino acids. Some amino acids function as neurotransmitters. We have 4 in particular that we know of: glutamate, aspartate, glycine, and GABA. Glutamate is an excitatory neurotransmitter. Glycine and GABA are known to be inhibitory. Most of our inhibitory neurotransmitters make it harder to reach threshold. Well, no, what the inhibitory neurotransmitter does is it makes it more difficult to reach threshold. So, for example, we know, like, if we hyperpolarize the membrane of a neuron, we push it further away from threshold. Threshold's minus 55. Resting potential of a neuron is minus 70. We get pushed down when we're becoming hyperpolarized down to at least minus 90. Most of your inhibitory neurotransmitters are going to take the membrane further away from threshold. So it makes it harder to get an action potential. Now the peptides. Peptides are just a type of protein because proteins are held together with peptide bonds. They're just amino acids linked together. We have 3 of them. Substance P, which helps us deal with pain. It does not block pain. We never block pain unless we use an anesthetic. And so we can't block pain in our bodies, but we can push it to the side. So it's still there, but we're not constantly thinking about it. And that's what substance P does. It allows us to mediate the pain. Endorphins are a natural opiate that makes us reduce the perception of pain. It doesn't block the pain signal, but it doesn't seem to be perceived as strong as it really is. So our bodies adapt a little bit to it. It's sort of like Tylenol. Tylenol doesn't do anything with pain except block the signals. Aspirin and Advil work by reducing inflammation, which reduces pain. Tylenol just blocks the signal. And that's what endorphins do. They help us to not perceive that the pain is as strong as it can be. The endorphins can also be a pleasure, somewhat of a pleasure neurotransmitter too. If you like chocolate, chocolate causes the release of endorphins. And so people that really like chocolate will eat chocolate because it tastes good. Because you're releasing, you get people eat like white chocolate, dark chocolate, milk chocolate, and it tastes good. They feel good when they eat this. It's because of the release of the endorphins. Now, we have some other peptides here involved in digestion, somatostatin and cholecystokinin. Cholecystokinin is the neurotransmitter that triggers the gallbladder to release bile into the small intestine to chop up fat. The presence of bile in the small intestine, and it's in this area in the duodenum, the first 10 inches or so of the small intestine. We'll talk all about this in AP2, is where fat gets chopped up and absorbed. Cholecystokinin is released when when lipids are in the small intestine, and that tells the gallbladder to release bile, tells the liver to continue making bile. Liver makes bile, not the gallbladder. Gallbladder just stores it. And so then we can chop up our fats. And plus we get to say cholecystokinin, which is sort of a neat word to say, so. Other neurotransmitters, there are gases. Nitric oxide. Nitric oxide is a very important vasodilator. It regulates, it gives us a localized regulation of the diameter of our blood vessel. And so it also keeps platelets and other structures from sticking inside our vessels. So nitric oxide, very, very important. We use nitric oxide in tablet form as nitroglycerin. Your patient that has chest pain, we call it angina, will pop a nitroglycerin tablet and they'll get a systemic body-wide vasodilation, and their blood pressure will drop, blood flow will increase, and pain will go away. So we also use nitric oxide in our ability to lay down new memories. Okay, so there's a lot of things our neurotransmitters do for us. We look at them as either excitatory, however, they're always going to be one or the other. They're going to either be excitatory or inhibitory. They're going to be excitatory or inhibitory. If they're excitatory, they allow us to depolarize. Now, there's no guarantee we're going to hit threshold when we depolarize, but we at least are moving in that direction. If we're inhibitory, we are hyperpolarizing. The charge in the membrane is going further and further away from threshold. So neurotransmitters, their effects are either depolarizing or hyperpolarizing. It's one or the other. Now, the same neurotransmitter, depending on the receptor sites, can do both. We know that the effect of certain of these amino acid neurotransmitters it depends on the receptor. GABA and glycine are inhibitory. GABA is a sedative that you administer a patient is going to make them sleepy and relaxed because it's going to enhance the role of GABA. GABA is driving the charge on the membrane of the neurons further away from threshold, so they're less likely to respond to a stimulus. So the same with glycine. Glutamate is excitatory. Glutamate stimulates the neuron to hit threshold. We find that when the brain is damaged, it releases lots of glutamate, and that causes that glutamate acts on many neurons, causing them to get more excited and release more glutamate, which kills more neurons. So it's sort of like a, like a wildfire, if you will, the way it spreads. So the effects and the actions are going to be different depending on what the receptors are. Now, as I said, acetylcholine is excitatory at the neuromuscular junction. It's inhibitory in cardiac muscle. Norepinephrine, released by the adrenal medulla, norepinephrine can be excitatory and inhibitory. Norepinephrine can cause skeletal muscle, smooth muscle to constrict, narrowing our blood vessels, but it also tells smooth muscle around our airway to relax. A person with stung by a bee that is caught, and the response of the bee sting is triggering vasodilation thanks to the histamine, and is experiencing vasodilation. Blood vessels dilate, blood pressure drops, plasma leaks out in tissues, they swell up from the bee sting. Okay, that's one consequence right there. It's, if not controlled, it can be fatal. The other thing that happens is that the same release of histamine causes a smooth muscle around the airway to start constricting. So now your patient is not only, their blood pressure is dropping, they're swelling up like a balloon, and they can't breathe because their airway is shutting down. Epinephrine, or rather norepinephrine, causes the blood vessels to constrict again, bringing the blood pressure back up, but tells the muscles around the airway to relax so they can breathe again. So your patient uses an EpiPen, or maybe they have a rescue inhaler or something like that. So anyway, let's see. Okay, so let's go on now. Now this is shifting gears completely from neurotransmitters, but when we have a postsynaptic neuron getting excited, that's the downstream neuron. We are sending information from the we have a presynaptic neuron depolarizing the dendrites of the postsynaptic neuron. We hit threshold, we have a new action potential running on down, maybe to another neuron, maybe to a muscle cell, maybe to a gland. If we're going to another neuron, we're continuing to process information. Remember, we have billions and billions of neurons in the central nervous system. 99% of all these neurons are going to be association neurons or integration neurons. They are the processing center of our brain. The ability to put together a comprehensive picture of what's going on depends on communicating to all these billions and billions of neurons that we use for integration of information. Like we look at the clock, to understand what time it is, we have to know, first of all, we have to understand the concept of time. We have to understand what a clock is. We have to understand what the numbers mean. We have to understand what the movement of the hands are. We have to know what hands are. And then we have to fit that into our schedule. All that has to happen for us to have to comprehend time and what we're doing. It isn't just, oh, look at the clock. We have to integrate all that information. We have to pull the information up from different parts of our brain so we know what's going on. So we integrate this information. How do you know what a phone is? How do you know what a banana is? It's yellow. How do you know what yellow is? Our eyeballs have no idea what the light rays that are coming off of these things mean. The eyeballs have no idea what yellow is. They don't know it's banana. However, parts of your brain back here in the occipital lobe can tell you exactly what it is, as long as all the light rays hit the right area and we put together a comprehensive picture of a banana that's yellow. It hasn't been eaten yet. It might be my lunch, So now, That's what integration is. And the way we put this information together is we operate in these groups of neurons. They form pools of neuron that take information from all different areas of the brain and tell us what we're seeing or hearing or smelling or tasting or feeling. You know, all of this information, all of this processing is taking place faster than we can comprehend, telling us what we're seeing, what we're hearing, what's touching our skin, you know, or is it too bright in here? It is awful bright in here today. You know, so is it raining? Is it dry? What's rain? What's dry? You know, what's the thing with all the stuff on it? What's stuff? We get a comprehensive picture from everything we see and hear and taste. Now, the pools are organized. Nothing in the brain is disorganized. You know, we have, in this case, you're looking at a presynaptic neuron coming down, and it's going to synapse at every one of its axon terminals. Now, you know that the multipolar neurons can synapse up to 10,000 times. And every one of those synapses with, you know, every one of the 10,000 synapses can themselves then synapse 10,000 more times. So the closer we are to the presynaptic neuron when we synapse, the more likely we're going to generate an action potential. You know, not everything is lined up in a nice straight line on, you know, it isn't like everybody's receiving the signal, you know, right in the front row. You know, we have neurons that are distributed around here because we may have to go, you know, maybe we're not connecting to all 10,000 right in front of us. Maybe we're connecting to 10,000, you know, neurons that are scattered all over the place. And so there's a little bit of a lag. The further away you are from the presynaptic neuron, the less likely you are to hit threshold. You know, maybe the important information is being distributed to the front. So we have a discharge zone. The neurons, you know, the brain is figuring out which neurons are important for this signal. Yeah, we're going to send it out to all 10,000 axon terminals, but let's do these here in the front first because they're important. Well, maybe the front later on is over here or up here or down here. It all depends on what the signal is. So now, when we process information, we use two ways. If you have to go to your advisor, okay, so it's fine. So when you process information, we run it through two mechanisms. So now I'm going to give everybody the answers to the test now that he's leaving. You can hear that. So, okay, you can tell him that in the lab today. So, okay, serial processing is like a set of Christmas lights. We've all seen Christmas tree lights where you break one bulb and the whole strand's ruined, right? We all know that. We've also seen the kind where you break a bulb and it doesn't matter and it continues to work fine. Serial processing is like the first example. When a bulb breaks on a set of Christmas lights, it's done. Serial processing, there's only one connection it's an all or none connection. You know, it's like wiring. You know, if you break the circuit anywhere, the whole circuit shuts down. Now, we do use serial processing in our brain and in our spinal cord. Yeah. The beauty of the serial processing is it's an all or none response. It either does or doesn't go. Reflexes, spinal reflexes are serial processing. We have a sensory receptor, we have a sensory neuron, we have a processing center, the integration center in the spinal cord, we have a motor output, and we have an effector. And nothing gets in the way of that signal. There are no extraneous signals coming in on a reflex. Reflexes are fast. Reflexes are predictable. We're always going to get the same response on a reflex. It's always predictable. You know, if you hit somebody in the patellar ligament properly, their leg's going to kick out. When you put your hand down on something hot, you're going to yank it back. That is a predictable reflex. We always do that. And this is serial processing. The reflex arc, nothing interferes with this. If we break any part of the chain, we don't get the reflex. If the sensory neuron is cut, we don't get the signals of the brain. If we damage the motor neuron, we don't get the signal out. I'm not going to remember the spinal cord. We don't get the signal out if the motor neuron's damaged. If we cut any part of the arc, then there's no signal. This is serial processing. It's fast. It's very fast because we don't have extraneous information floating around. But everything has to work. Parallel processing is a little more sophisticated. Parallel processing is the Christmas light example where you break a bulb and all the rest of the lights still work because you have a double wiring system. Here with parallel processing, we have multiple pathways. And so we can have, we're not limited to just a pathway here. We use this for more higher level processing. Consider if you smell an odor. I guess you would always smell an odor, but you detect an odor. Now, some odor you can immediately detect, is that a pleasant odor? Is that an unpleasant odor? If I were to peel the banana and hold it up underneath your nose, you'd know it was a banana. You'd recognize the odor. Not only do you stimulate your chemoreceptors in your nasal cavity, and you stimulate the pathways in the olfactory bulb leading to the olfactory tract, leading to the temporal lobe where we store all the memories of odors and tell you that's a banana, but you may be able to detect, is that a ripe banana or is that an overripe banana? There's a whole discussion. Do you like green bananas or do you like brown bananas? Yeah, I know. In the four of us in natural sciences up here, we're split right down the middle. Dr. Grice and I are green banana people, and Dr. Malik and Evan are brown bananas. You're green?
I'm with him.
You're with him, okay. And Jillian is a brown banana person too. So Dr. Grice and I are sort of just holding the line with our green bananas. So why? It's because of, and we can tell the difference because of parallel processing. Not the texture of the banana, the odor of the banana, the flavor of the banana is going to run down through multiple pathways to give us that comprehensive picture. That is what parallel processing does. It's just not one signal. It's multiple signals on taste and texture and odor and the mushiness of the banana, if you will, all that. So all that's going to come together to give us a comprehensive picture. Furthermore, odors and memories are really, really tied together. A lot of emotional memories are triggered by odors. Flowers, food, cologne, are all going to trigger different types of memories. Flavors of food, you expect food to taste a certain way when you smell it. If you smell barbecue cooking, you expect it to have a certain flavor to it. Baking bread, you expect the bread to have a certain taste to it. Certain flowers at different times of the year evoke memories of previous events, for example. It isn't just a one loop type of thing with parallel processing. Now, when we do parallel processing, we find 4 different types of circuits involved here: diverging circuits, converging circuits, reverberating circuits, and what we call the parallel after discharge. This is a diverging circuit. One neuron stimulates a whole bunch. or more properly, it's an amplifying circuit. Every time we move a muscle, a skeletal muscle, that's a diverging circuit. One motor nerve stimulates many motor neurons, which stimulate many axon terminals, which stimulate many muscle cells. One nerve, many neurons, even more muscle cells. So the motor unit, for example, a motor unit, one nerve can stimulate 300 cells. That's a diverging circuit. A converging circuit is where you have multiple input, all reaching the same goal. Get back to the banana. Odor, texture, taste, flavor, all come together to say it's a banana, it's overripe, or it's underripe. It tastes, it doesn't taste right. And there's a reason why people like the overripe bananas that are brown, because the longer the material, whatever that we call the material inside the banana ripens, it gets sweeter. More and more the starches and carbohydrates there break down into more simple sugars, and so it has a sweeter flavor. So even though it looks disgusting, but it has a sweeter flavor, which is why we like, some people like them overripe, and those of us that are normal like them green. So yeah, that's my take. They're great for banana bread. So anybody else is just wrong. So not really. So okay, that is a converging circuit. Lots of input giving you one comprehensive picture. Lots of input giving you a comprehensive picture. So it triggers the same thing. You get a memory, for example, based on odors and sounds and smell and taste, and all these things come together. The reverberating circuit, or we often call it an oscillating circuit, is something that happens over and over and over again. We have to think about it. We breathe every 4 seconds, 15 times a minute. We don't have to stop and think to breathe. The signal to breathe loops back on itself. It comes down through these neurons and loops back on itself and tells us to do it again. That's a reverberating circuit. When we walk, if you move your arms when you walk, that is a reverberating circuit. If you walk normally, or when you're walking normally, you tend to put your toe goes down, then your heel goes down, then your toes come up, and your heel comes up. We walk heel to toe. Now, we don't stop to think about it, because if we did, we'd fall down. If you ever try to watch yourself walk, it's not going to end well. So I know.
What if you have to, though?
Well, yeah, I mean, if you have to watch it, but see, that's different. If you're watching where you're going, that's different. But if you're actually watching how I'm lifting up my foot, and don't ever try it on the stairs. I'll just tell you that. Don't ever try it on when you're going up or down a flight of stairs. Things that we do frequently that we want to have the same response every time is a reverberating circuit. We don't think about how we walk, but there's a lot of things going on here. Our toes have to spread out when our toes go down, when our foot goes down, our toes have to spread out to stabilize our position. Then our heel has to go down so our toes come up so we can take the next step, and we go back and forth like that. And so we have a lot of muscles at play here, and we're not thinking about it because it's a repeating circuit. And it's a good thing, because it would be, if we had to stop and think about how we walk, we'd never get anything else done. So, and then the last type here, the parallel after discharge circuit. We have multiple pathways leading to one output. Now, what does this mean? Well, you ever get a package from, somewhere that you have to assemble the parts to make something like a bookcase or a cabinet or whatever. Everything these days comes in pieces instead of coming as one complete thing. So you have to put it together. And they give you the instructions either written or in pictures. I love pictures. I'm a very visual person. I love putting stuff together with pictures. But you ever have that where it doesn't seem to go right because the picture looks like it's upside down and backwards, and you can't make any sense out of the picture? And you look at it and you look at it, and your brain is processing it, and all of a sudden the little light bulb goes off. And oh, you turn it over, and that's exactly how it's supposed to fit. It's that aha, that light bulb moment you get when you're working through a problem and you can't figure it out, and suddenly there it is, right there. Because these pathways run at different speeds. And so you're running the same information through a variety of neurons, and some are faster than others. And when they all hit, when the ones that are lagging behind hit the right location, you have that sudden insight. Doing, if you're taking Providence stats right now, I think we're taking Providence stats, or you will at some point, I'm sure. Don't shake your head, you will. So if you've had a math class somewhere in the past, and you don't get it, and all of a sudden you get it, you understand it. You have that sudden burst of, oh, that's how it works. So you understand better now. So, and that's what this parallel after discharge circuit does. Very complex problems that you've had to process for a while. Many times this happens while you're asleep when we're dealing with our subconscious. If you're thinking, this is the kind of stuff that happens when you're cutting the grass, and suddenly you get this idea in the middle of it, and you're just busy trying to cut straight lines, and all of a sudden, hey, there's an idea, because you're not doing anything else. Your brain is sort of into neutral when you're just working in the yard. Lots of scientific breakthroughs have occurred when an individual has discovered something, has woken up in the middle of the night, and suddenly they had just dreamed the answer to the problem and wrote it down. So, okay, and that is the end of chapter 11. So let me bring up chapter 12 now. We're going to be looking at the brain. So let me make sure that I am sharing this. Okay. So, okay. This is the second chapter in Unit 4. This is going to look at the brain and the spinal cord, with most of the focus on the brain, but we'll get there. So, okay. The brain and spinal cord. The brain is about the size of it weighs about 3 pounds. Let me give you an idea just how big a human brain is. This is I want to show you a life-size model right here. And you can see that I'm holding it in my hand. So this is about the size of a human brain. This is, this replica in here. So this is actually much heavier than a human brain. The human brain only weighs about 3 pounds. It has the consistency of Jell-O or maybe oatmeal, because it's very squishy, and it does not support its own weight. Back here. The brain is, male brain weighs about 1,600 grams. The female brain is about 1,450 grams. Doesn't matter because the sizes are different because they are equivalent. The average body stature is a little bit different. Males have a slightly larger body stature than females do, but so the brains are correspondingly bigger. But the actual structure is equivalent. There is no correlation, let me get that out here right now, between the size of the brain and intelligence. A large brain doesn't mean that a person is smarter than someone with a smaller brain. So size doesn't matter when dealing with intelligence. There is no correlation with race and intelligence or sex and intelligence. It has nothing to do with any of those. Intelligence is sort of one of those hard to grasp ideas that are variable, but it has nothing to do with any of those physical characteristics. Now, our brain allows us to be irritated, not in the way we think. Irritation means responding to sensations. We are irritable. That means we respond to a stimulus. Doesn't mean we're unpleasant. So we deal with sensations. We respond to sensations. We are aware of sensations. We have, the magic word is perception. Perception of sensations. We have memory. We store lots and lots and lots of memories in our brain. We can go back to maybe when we have isolated memories that still pop up when we're 2 or 3 years old. That's probably the earliest we can go back, and then there aren't that many that early in life. But we remember all sorts of things. We know names and dates and places and faces and historical facts and scientific facts. We remember how to drive. We remember how to dress. We remember what we like to eat and what we don't like to eat. So we store all this stuff in memory. We have emotions. We have an emotional brain and a logical brain. They're at war with each other. Sometimes the emotional side wins and sometimes the logical side wins. Usually it's a draw. We make decisions, some good, some bad, whereas some not so good in here. And our behavior is dictated by our brain, partly by our emotions, partly by our logic. We know what is right and wrong. We learn that. We learn fairness very early in life. So we know all about, well, we're usually taught right and wrong. And it is through learning. We establish that behavior pattern. So now, parts of the brain. The large hemispheres are called the cerebral hemispheres. That's these things. That's these things. Boy, I'm glad this isn't an English class. Okay. These are called the hemispheres. This is, show you this one. This is a hemisphere, one side of the brain, the cerebral, we call it the cerebral hemispheres on our brain. These large structures that set, come on camera, there we go. These large structures that set on top right there, these are the hemispheres. That's where we are located. That's where our personality is located, where most of our decision-making and intelligence are located. Everything else underneath that is all about integration and processing. So we have the cerebral hemispheres. We have what's known as the diencephalon. Diencephalon is this structure right in here. It looks like, later on you'll see it looks like a small egg, and they're paired. This is called the thalamus. All sensory input comes into the thalamus and then gets routed out to its appropriate area to be processed. The hypothalamus is the link between the nervous system and our hormones. It's between the other control mechanism. The nervous system is the #1 control mechanism. The endocrine system is our number 2 control mechanism, and the link between the two is the hypothalamus. It's also our body's thermostat. It regulates our body temperature, and that is the diencephalon. At the base of the brain is the structure known as the cerebellum. This is our cerebellum. It is the second largest area there we go, right here. This is our cerebellum here, right at the base of the cerebral hemispheres. And they're paired one on each side. The cerebellum is where we store our motor memory. It's how we remember to do things, the areas that improve with practice. It's also where we regulate our equilibrium and our balance. So the cerebellum plays a big role in walking and talking and riding a bicycle and driving and things like this. And then the last part is the brainstem. These things highlighted in green. The brainstem consists of a structure known as the midbrain, which is this area right here. It looks like a bird. And then we have what's known as the pons. This green oval is the pons. And then the last part of the brainstem is called the medulla oblongata. And from there, we run directly into the spinal cord. And where the medulla ends and the spinal cord begins is sort of problematic because, it just sort of runs together on that. So our brain is covered by the skull. We have the outer layer of the skull, of course, is the bone, and the periosteal layer underneath the bone. We have 3 layers of connective tissue known as the meninges that wrap around the brain and the spinal cord. And these are the meninges. Here, oops, I got carried away. Hang on a second. There's the bone. There's the periosteum layer. Underneath that is what's known as the dura mater. Then we have the arachnoid layer or arachnoid mater. And then we have the pia mater. And the dura mater is a tough layer of connective tissue, and it surrounds the brain, it surrounds the spinal cord. And underneath that is the intermediate layer, the arachnoid layer, is where we find our blood vessels. And then the pia mater is glued to the surface of the brain. And in between those layers, we have about 6 ounces of cerebrospinal fluid filling in all the holes in our head. We have lots of cavities in our brain and running down the spinal cord so the brain can float in this sack of salt water. Because the brain can't support its own weight. So it will float inside the meninges. It functions as a shock absorber. When we turn our head suddenly, or when we get hit in the head, our brain bounces around, sloshes around on the inside of the skull, but it isn't damaged usually because all that fluid in there, well, it doesn't sound like very much, is enough to absorb most shock. So, and the meninges are anchored to the crista galli of the ethmoid bone, remember that thing that sticks up right in the middle of the skull. You got the cribriform plate on either side of it with little holes for the olfactory nerves, and that little bump there at the crista galli. That's the anchor of the meninges. So the brain doesn't go sliding around too awful much in there, and it's still floating inside the sac. And so we have the meninges, what they're called. We have the dura mater, that's the outer layer, the arachnoid layer, and we have the pia mater. The meninges can become infected. Inflammation of the meninges, either bacterial or viral, is called meningitis. You've probably all heard that term before. Very contagious disease, but it affects the brain because it's called, and we have inflammation, and with inflammation you have swelling, and our brain does not like to swell. There's no place for it to swell to. So, okay, now the brain is supplied by a great deal of blood. We run the carotid arteries take blood to the brain. We have various cerebral arteries that run through the brain. The supply is redundant, meaning we have lots of connections going to the same place. The heart's the same way, where we're not limited to just one large vessel in the heart. We wrap the capillaries, because capillaries are where the exchange of nutrients and oxygen are going to take place. The capillaries are the smallest of the vessels, and the only place where we get exchange of gases and exchange of nutrients and waste is at the capillary level. Now, we have the astrocytes. Remember the astrocytes? They wrap themselves around the capillaries. They form this thing called the blood-brain barrier. And that's to protect the blood so that, and protect the brain so that contaminants in the blood can't get into the brain, and that any kind of contaminants in the brain can't get into the blood. The only thing that can come through the blood-brain barrier is oxygen going out, carbon dioxide coming in, glucose going out, waste products coming in, nothing else. Of course, alcohol can go across the blood-brain barrier into the brain. Various anesthetics work, but we have a problem with other materials going across this barrier. The capillaries are held together, wrapped up by the astrocytes, and then we have these cavities formed by the ependymal cells. The ependymal cells generate, that's the green things, they generate and circulate the cerebrospinal fluid. And so the ependymal cells are, with having tight junctions, are very, very particular in what comes in or goes out. And the astrocytes, with the astrocytes wrapping around the capillaries and the ependymal cells held together with tight junctions, stuff doesn't go into the brain that the glial cells don't want. But we can allow glucose and ions and oxygen to leave the blood and go into the cells, and we can allow waste products to leave the cells and go back into the blood. And so we have a very challenging barrier here, the blood-brain barrier. It does a really good job protecting the brain, but it makes it harder to treat infections, for example, in the brain, because medications that might help an infection or inflammation can't get across. Now, the spinal fluid is only about, we talk about it, but there's only about 5 ounces of spinal fluid, cerebrospinal fluid, and that includes what has to go down around the spinal cord itself. But remember, the brain is a pretty snug fit inside the skull, so there isn't a whole lot of space here, but we have a number of ventricles, cavities inside the brain. And so that 5 ounces fits around everything pretty well. It allows the brain to float because the brain can't support itself. It has to float. It can't support its own weight. It functions as a shock absorber, so that when our brain does move suddenly from side to side or front to back, it's going to impact first against the cerebrospinal fluid, and whatever impact it was going to have isn't as bad. The spinal fluid maintains the right sodium concentration around the brain cells. Remember, we are dealing with neurons that have to have sodium on the outside and potassium on the inside. And so the sodium and potassium in the spinal fluid, cerebrospinal fluid, allow us to maintain that potential. And then, of course, it helps us get rid of waste products too. So, okay. Enough about cerebrospinal fluid. Let's talk about the brainstem. The brainstem is like a stalk that holds the cerebral hemispheres of it. It's like a mushroom. If you look at this model here, this is the brainstem. here's the cerebral hemispheres right here. This area here is the brainstem with the medulla, the pons, and the midbrain right in here. And so it's, you know, think of a mushroom. Think of a mushroom holding up the brain, the stem of a mushroom, and that's what the brain stem is for us. And now let's get back to this. So the parts of the brain stem are the midbrain, the pons, and the medulla oblongata. Information from the spinal cord comes up into the brainstem. Information from the brain goes down the spinal cord from the brainstem. It has some very important reflex centers in the brainstem. In the medulla oblongata, we control our breathing. We control swallowing. Remember the hypoglossal nerve? Remember the hypoglossal canal, a little tiny hole on either side of the foramen magnum inside the skull. Hypoglossal nerve goes up to the medulla oblongata, the lowest part of the brainstem. Hypoglossal nerve innervates the tongue so that we can swallow. With damage to the brainstem makes it impossible. Damage to the medulla makes it impossible for your patient to swallow. And so they're in a world of hurt here. And they end up having they end up aspirating food and water into their lungs. We regulate our heart rate with the medulla in the brainstem. We don't initiate the heart rate, that's done at the heart, but our ability to speed up our heart rate or slow it down occurs in the medulla. Same with blood pressure and consciousness. Our ability to be awake or to ignore outside stimulus, it doesn't matter, comes through the brainstem too. And so while it doesn't seem like it's a very large structure, it plays a big role in maintaining our day-to-day activities. So first thing we see is the midbrain. There's two parts to it. There's a ventral part and a dorsal part. The midbrain is this area in green that looks like a bird, if you imagine that. That's the ventral part of the midbrain. And this other part back here is the dorsal midbrain. So, and right in between them is what we call the cerebral aqueduct. It's a passage for cerebrospinal fluid. And there are two ventricles here. Ventricles are holes in the brain. The third ventricle surrounds this blue structure here. It looks like an egg. That is the thalamus. The third ventricle surrounds the thalamus, covers it with cerebrospinal fluid, and that spinal fluid moves down through the cerebral aqueduct here to the fourth ventricle, this structure here right underneath the cerebellum. Now the midbrain consists of this structure here that's shaped like a bird and this area behind the aqueduct. So you have on the ventral surface, the side facing forward of the brain on the belly side, if you will, you've got three clusters of three structures here. We have what are known as the cerebral peduncles. That they're outlined here in blue. Get this out of the way here. There we go. These are the cerebral peduncles here. We are facing forward now. Cerebral peduncles, they look like they're holding up the brain, but they're not. What they are both sensory and motor input. Sensory input, motor output. For every sensory nerve you're going to have, you got to have a motor nerve to balance it. And so we have a lot of sensory and motor activity right there. We have a dark area, a darkly stained area called the substantia nigra. This has a high content of what we call melatonin, the pigment molecule. Melatonin determines our in the skin determines our color. Here we have large concentrations of melatonin in the substantia nigra. The substantia nigra appears dark because there's lots of melatonin there. Melatonin is the precursor for dopamine. We make melatonin, and then we can use that to make dopamine. Dopamine isn't just about pleasure, it's also about motor control. And then we have the red nucleus. As we move a little further back in here, the red nucleus is red because it contains large concentrations of iron. And we send information from the cortex and the cerebellum through the red nucleus. Now, why is the cortex and cerebellum involved? Because motor activity originates in the outer covering of the cerebral hemispheres. We call that the cortex. Motor activity starts high up in the brain, in the area called the cerebral cortex. The cerebellum takes those raw signals and says, well, this is the best way to do it. The cerebellum refines the signal and says, well, if you're going to throw the banana at somebody in class, don't, twirl it around like this because the stem will break off. The cerebellum tells you the best way to do something. We don't have to stop and think about it. All we say, I want to throw the banana at somebody, and the cerebellum will tell me the best way to do it. It's all instantaneous. We depend on the position of our muscles, which the cerebellum knows. We think about our cerebellum remembers the best way to do it from previous experiences. Not that I throw a lot of bananas, but how do you know I haven't been practicing? And so we get the most efficient use of our muscles thanks to the cerebellum. And so the information telling us how to send the signal goes back and forth through the red nucleus. Now, the substantia nigra has a very important role to play. Of all the areas, this is probably the most important. There is another structure called the basal ganglia. The basal ganglia is made up of a variety of structures here, including the substantia nigra. And what the basal ganglia does is when our cerebral cortex tells us to do something, then the signal will come down into the basal ganglia, and then the basal ganglia will tell which muscles to use and which muscles not to use. Because when the substantia nigra goes to work, it will stimulate using dopamine certain excitatory muscles, and it will inhibit other muscles, because we always have antagonistic types, clusters of muscles. Like our hamstrings are the antagonists of the quads. If you're going to kick something with your foot, you don't want the hamstrings getting in the way if you're going to use your quads. And so when we decide we're going to do something, when we come up with an action from the cerebral cortex, the us part of our brain, we have to run it through the basal ganglia. So we use the right muscles, because we got a whole bunch of muscles. Everybody wants to do something. We have to control the ones that are not going to be contributing to the action, because we want our action to be smooth and simple and effective. The basal ganglia lets us plan our action. I want to pick up my pencil. So I pick up my pencil and I put it down. The basal ganglia allows us to stimulate the muscles necessary to pick the pencil up and inhibit the muscles that would get in the way. Because there's lots of muscles that might also be stimulated. If I'm going to pick up my pencil I don't want my arm to be flexing at the same time as I'm trying to pick up my pencil, because that would make it very challenging. So I inhibit the muscles that would cause flexing or extension. I only use the muscles I need to use, and we get a nice fluid motion. We've planned it. We don't think about it, but we're planning it. You know, I want to pick up my pencil. I'm going to pick up this rubber covering on the ocular and put it back down. We plan all these out instantly, and we have a fluid motion. We don't have a sudden erratic type of grabbing at a structure trying to pick it up or do something with it. Now, how does the substantia nigra do this? How does it help the basal ganglia? The two pathways in the basal ganglia, one is excitatory, one is inhibitory. Dopamine moderates the inhibitory pathway. It keeps the inhibitory pathway in check so that the only actions that we do are the ones we want to do. You know, I want to pick up your phone. I want to put your phone down. I want to pick up my coffee cup. I want to put that down. I don't want to go knocking it over or knock over the keyboard trying to pick up my coffee. Dopamine blocks any antagonistic actions that may get in the way. Now, where does this, why am I talking about this? Where does this come into play? There is a disease known as Parkinson's. In Parkinson's disease, the substantia nigra breaks down. It doesn't release dopamine. It doesn't hold the inhibitory, it doesn't hold the antagonistic muscles in check. And it turns out that if we don't have any dopamine, not only do we not hold the antagonistic muscles in check, but they become the dominant response. So a person with Parkinson's disease has trouble walking, they have trouble talking. Their face is usually paralyzed. They have a tremor, they shake constantly. Their muscle tone rises. You know, remember what muscle tone is? Muscle tone is we rotate a stimulus around the motor unit. So we have a motor unit fire and it warms up the muscle cells. And we have another one fire and another one fire. But what happens here is we don't rotate. We start raising the muscle tone. And so your patient with Parkinson's has a great deal of difficulty in simple tasks like, you know, like walking and talking and movement, because the muscle tone levels are increasing in the antagonistic muscles. I want to pick up my coffee cup, and yet my hand is, I have a tremor that I can't control. And because it's the skeletal muscles are fighting. Skeletal muscles are fighting the antagonistic muscles. The antagonistic muscles are winning because they would normally be kept in check with the dopamine. So this is Parkinson's disease. We know what causes it. Getting it to treat it, to find an effective treatment for it is the challenge. So anyway, now on the back side of the midbrain, we have a structure called the corpora quadrigemina. And there are two bumps. There are actually four bumps here, two on top of the other two. We have what's known as the superior colliculi, two raised bumps just below the thalamus. You can see where the superior colliculi is here. It says red circles. Below that is the inferior colliculi. Superior because it's on top, inferior because it's on the bottom. The superior colliculi is a reflex center for vision. It's a reflex center for vision. If I were to throw something at you and you caught it, because that would be a reflex to suddenly throw your hand up and catch it in the air. You tracked it, you saw it, and you responded. That's a reflex. You turn your head when you simulate your peripheral vision. You're driving down the road and you sense motion on the side outside your car, and it can be a deer, it could be a dog, it could be a kid, it could be a tree falling over, it could be anything. Peripheral vision stimulates the corpora quadrigemina's superior colliculi and you respond reflexively. A bright light causes you to turn your head in the direction of the bright light. So that is a reflex center for vision. The inferior colliculus is a reflex center for sound. You hear a noise, you turn your head in the direction of the noise. You're driving down the road and you hear a siren. You don't know if it's police or the fire or whatever, and you're looking around trying to see what's going on. Or someone beeps their horn at you. Now they could be 3 cars back, but you think it's the person directly behind you. We always do, you know. So it's very rarely the person behind you. Sometimes it is, but you turn around at the sound. Someone calls your name, and you turn in the direction where you heard it. That's a reflex. So these are 2 very important reflex centers in our brain: the superior colliculus and the inferior colliculus. In lab this afternoon, I'm going to show you exactly where they are found in the brain. They're directly below the occipital lobes and directly above the cerebellum. They're very easy to identify. So, and they look something like this. Here's a brain. There's the cerebral hemispheres there. This is the occipital lobe. There's the superior colliculus. There's the inferior colliculus right there. They are the startle reflexes: a sudden motion or movement, or a sudden sound. Peripheral vision. The rods detect, peripheral vision. And so it's something I say, out of the corner of my eye, I saw it. Corners, but you know what I mean. So these are visual reflex centers. We also use the superior colliculus, when we're assessing motion. You're sitting at an intersection and you're trying to pull out because there's lots of traffic going both ways, and you're trying to pull out, and you look for a gap in traffic because you assess how far a car is in either direction and how fast you can pull out. And you don't pull out right in front of a car, and hopefully you don't there's nothing moving for 5 miles in each direction, and then you pull out. So okay, and that's a good place to stop for now. So any questions on anything? Okay, then I'm going to get us out of here.